Welcome back. Before lunch, we met Malin Elfstrand, who talked about increasing knowledge regarding disease resistance among trees in order to improve forest health. And now it's time to learn more about cell shape, the importance of the cell wall structure, and how it adapts to change. So please warmly welcome Professor Stéphanie Robert.
I became a researcher as I'm curious and I like challenges. The idea that I could bring more knowledge in the field of science was exciting for me. I'm also a social person, and discussing new ideas with diverse colleagues from all over the world not only brings me motivation and energy, but makes me humble and willing to know more. My name is Stéphanie Robert, and I'm a professor in plant physiology. So my main research interest is understanding how plants develop by forming different cell shapes. So plant cells come with a variety of shapes and function, and they have to coordinate their growth in response to the environment to give the plant its final structure. This triggers my curiosity the most, how the cell develop at the good place in the good time. We already showed that the cell wall, a structure that surrounds each plant cell, and that glue cells together, has a true function in controlling cell shape. There is also a protective layer of waxes covering the above-ground parts of the plant, and it's called the cuticle. And we are now investigating whether the cuticle has a function in controlling cell shape. My curiosity motivates my research. I aim to improve our understanding of how plants grow and develop. I'm aiming to acquire basic knowledge and make this knowledge available to inspire application for the society. Enhancing our knowledge in this area is necessary for future improvement in agriculture and forestry.
Welcome, Stéphanie.
Thank you.
I know you're a strong advocate of basic research. Why is basic research so important?
I believe that if we bring basic knowledge, so if we try to understand how things work, and in our case, how plants develop, we will bring all this knowledge available for the public. And the public can use this to be creative and have a lot of ideas to develop around this. But my patient is ready to understand how things work. So I'm here to provide what is in the book in the library, to provide the basic knowledge.
Oh, nice metaphor. I know that the Umeå Plant Science Center, where you work, include people from all over the world. What does the diversity mean to research and to job satisfaction in your department?
So at Umeå Plant Science Center, I think we are around 47 nationalities for 250 people. So it's just, we live in a very diverse world, and it's fantastic because you have view from people bring their own view from their own background. You can adapt to the other. You can get so much from other people with different point of view. So they bring new ideas, new way of looking at things. Everybody comes with his own character, and it's. I really think it's fantastic.
It sounds like a very nice culture to be part of. Well, as I asked Malin Elfstrand about the same question before lunch, you seem very passionate about engaging in students. And how do their involvement contribute to your job satisfaction?
So one of my fuel, I would say, or motivation bringer is like raising new scientists. So especially in the lab, I like to form people to develop their own science in their own way with my leadership. I'm there to lead them. And it's fantastic to see the development over the years, especially with PhD students, for example. When they come, they are very naive and want to save the world, but you just, you manage to trigger them into the process. And when they come to the office with a new idea that I didn't even think about it, I'm like, okay, my job is done. They can go on. And this is this is the motivation.
So you get new insights from the students as
I
well.
get new insights from the students,
Okay.
but they also, it's the same. They have like different, like the new generation. I might seem old, but the new generation has a different way to look at things, and I think now it's really clear that I'm older, and it's really good to have their impact on the research.
What are the new perspectives? You say the new generation has new perspectives. What
Like all the way to with artificial intelligence, how to use that, how they put it in their way of analyzing the things is different. It's and it's good.
How exciting! Tell us more about your research. So please enter the stage. Yes, and I would like to introduce Stéphanie Robert, professor of plant physiology, talking about how plant cells take shape.
Thank you. So now I have the opportunity to show you a bit what my group is working on. And we are really focusing on plant cells take their shape. And I will explain you why it's important. So first of all, I would like to put myself as a clear defender of plant science. And I want to show people that plant science is important and why is it important. First, plants produce most of the oxygen we are breathing. So they take the carbon from the atmosphere and release oxygen by a phenomenon that is called photosynthesis. So thanks to plants, we can breathe. But they are also the base of a lot of things we are eating. And here you see a nice vegetable that we can grow in the north. And also all the fuel that we are using is based, a lot of the fuel we are using is based on plant. And also, if you look around you, all the furniture and everything is based on plant material. So we spoke about trees before this morning, and this is all based on plant material. But why I am fascinated by plants? I'm fascinated by plants because of their development. So the really nice thing about plants is that they can create new organs along their development. So if you are a human being, you are born with one head, 2 arms, 2 legs, and hopefully you will grow with all these organs along your life, but you don't change shape. However, if you are a plant, or a plant is growing, so you will have a seed that is germinating with 1 root, 1 stem, and over the development it will create more roots, more stem, more leaves, and this is called de novo organogenesis. And this is fantastic to understand this process. And I will explain you why. So this process is adapted to the environment the plants are growing in, because plants cannot run away like animals do, or we do. If we are cold, we put a new jacket, or we go to California, but plants cannot do that, right? So they have to adapt to the water availability around them, the nutrient availability, all the environmental stresses they have around, and they will shape, they will grow in function of this. And why is it so special? Is because you find plants all over the world. Wherever you go, you will find plants. If you go to the desert of Joshua Tree, you will find plants. If you go close to Umeå in Norrland, you will find plants. But you go to China, you will also find plants. They are different, they look different because they are adapting to their environment. And this is called developmental plasticity. So why is it important to understand this? Because we are facing climate change. We are facing a lot of changes that will come or that are already around. We have increasing ambient temperature. We have less water available. We have more soil salinity. So all of this will, of course, change the environment of all the plants around us, the plants we know. And we have to understand how the plant will adapt to that, to, of course, us as humanity to survive. So SLU one of the goals of SLU is to provide knowledge for reaching these sustainable goals of the United Nations. And I think by doing plant science, we can do that. We can provide some of the knowledge for these sustainable goals. For example, the sustainable goals number 2, zero hunger, is obvious, right? Because plant science will provide new ways to produce food, and of course to fight climate change with forests, etc. Plant science could also provide knowledge for that. So now if I come back to that, I tell you from the beginning that plants are adapting to the environment by making new organs. But what we do in my lab is that we zoom in on these organs, and every organs have different cells. So if you zoom in, you will see that every organ has different cell. Every cell has a different shape, and this shape is according to the function of the cell. So the cell has to take a shape at the good time and in the good place to be able to function, to build a proper organ, and to build a proper development. So now I would like to introduce Arabidopsis thaliana. So that's a Latin name. In English, it's tail cress, and in Swedish, pardon my Swedish, but it's backtrav. And this is a plant we are working on. It's a genetic model that we are using because of a lot of reasons, and including the genomic sequence for some years. So now if I take you slowly to what we do, is that we look at the cell shape, particularly in the leaf. So if you zoom in on the leaf, in this case of Arabidopsis, you will see different kind of cells with different kind of shape. And we are particularly interested in the cells that look like a puzzle. They are called pavement cells because they are like the pavement of the street, pavement stone of the street. And it's fascinating how they can grow because they are really interdigitated to each other. So they are called jigsaw puzzle shape because of this. They look like a puzzle. They are growing. One while is growing into the other one, they are acquiring their shape. So the coordination can be done within the cell, but also with the rest of the cell together. And why is it so important? It's important because it's contributing to tissue integrity. So the plant have a tissue that is solid enough to resist to the environmental stress, and it creates mechanical strength if you have wind or other things. And it also regulates for pathogens attacks and things like this. So like now on the left side of the slide, you have a picture of an Arabidopsis that doesn't have this tissue integrity, and you see that the tissue is actually broken, and this plant will soon die. So here, why we are looking at these cells, because the shape is nice and the shape acquisition is complex. And the way we do it is that we look at it over time. So here you have again this Arabidopsis thaliana plant leaves with different ages, so day 2, 4, and 7, so this is in time. And we can see that the development is, we can look at different cells that are here either not shaped, and after 7 days the cells are shaped. But you can also see that in this part of the leaf, the cells are less shaped than in this part of the leaves. So this is like a bit this Escher facade. So this is a painter, and some people paint it on a building in Spain, where here the shapes are very simple. They are square and almost square, and get more and more complex. So we have a way to look at this cell shape acquisition over one organ. So now I will take you back nicely to your biology textbook. And what is very different in plant cells is that they have a cell wall. So the cell wall is a network that is placed outside of the cell, outside of the plasma membrane, and composed of different sugar that we call polysaccharide. And the cell wall is extremely important because it's provide rigidity to the plant. The plant have no muscles, and for a tree to stand, you need to have the cell wall. But the cell wall has to be also flexible because it has to allow elongation of the cells as well. So this is a fascinating thing to look at. So now what is the connection between cell wall and plant cell shape acquisition? So that's, again, we go back to the cells that I showed you before, but if we remove the cell wall from the cells, you will lose completely the shape. The cells are now round, and there is no predetermined shape. So that shows us already that the cell wall is participating in the cell shape determination. So what we did is that we tried to understand a bit this, and we look at different genetically modified lines of Arabidopsis, and these lines had different cell wall composition, and we look at the cell shape, at the pavement cell shape on these lines. And we see that compared to the control, all these lines show differences in cell shape. So we could conclude that cell wall was important for the cell shape acquisition again. And then we use hardcore technology or new technology to understand that. So how the cell wall mechanochemical properties would determine the shape. And what we did is that we use a very cutting-edge technique and at very high resolution. It's called atomic force microscopy, and I will not go to details, but it's basically a tip that will tip on the surface, and if it's bouncing back, that means that it's like on a trampoline, right? It's very soft. And if it's bouncing back less, then that means that the material is hard. So I will not go to extreme details, but we show that the cell wall properties using this atomic force microscopy, we show that the cell wall mechanical properties were heterologous along and across the cell wall. That was the first time somebody was showing this. And we could correlate this with the composition, so the sugar composition were also not homogeneous in the cell wall. But just to create another to show you that multidisciplinarity is also important in the research we do, is that we use mathematical modeling with people that were at this time in Lund, and we show that this heterogeneity is necessary for the shape. Because if we have a cell wall, I will also not go to details, but if you have a cell wall that is homogeneous, everywhere the same, you will not get this wavy shape. So our group has shown and continue to work on this, that the cell wall contribute to cell shape acquisition. And I hope you understand that this basic knowledge on cell shape and cell wall and cell shape acquisition is very important for the sustainable goal. So how can scientists like us can contribute to that? The knowledge we produce can help to develop plants that are drought or stress tolerant. So if we have less water, we can still cultivate what we need to eat or live. We could also provide knowledge to go to a sustainable society with creating plants that need less fertilizer or water, that are resistant to pathogen, that are more nutritious, so we need less plants at the end. And we could also, of course, contribute to the green energy wave that is going on now. So I think it's a fantastic time to do science, and especially plant science, because people start to understand more and more why plants are essential in their life. I also think, as we discussed before with Malin, that curiosity-driven science is important, and we create a knowledge that is accessible to the public, and they can use that and do a lot of innovation. I believe in multidisciplinarity, in exchange with colleagues, and most importantly, in diversity. So I would like to thank you now for everything.
Thank you very much, Stephanie. Well, you mentioned in your last picture, when you showed your last picture, you mentioned that now is an exciting time for science. Could you elaborate on why you say that?
It's for so many reasons, but one of the reasons is technology. We get access to a lot of new technology that is giving us so much opportunity to change the resolution of our study, because we are cell biologists and we have to go closer and closer, and this is fantastic. And it's also like the word accessibility also, like it's so fast now, so it's good and bad, but it's very fast to do science now.
Okay. And I just want, just as a reminder, those of you in the audience, you're welcome with your questions. And if you're watching the broadcast, you can email your questions, and you can just, in the studio, you can just raise your hand and you will get a microphone. So feel free to ask questions. Well, can you, yeah, we have a question over there. Pernilla, please.
Thank you, Stephanie. So is it by manipulation that you can make these more resistant to pathogens like Malin talked about, and to drought and climate change overall?
It can be selection also. So
okay.
you can think about selection, so like the breeding and select plants. If you know which genes are providing a resistance, you could select for this kind of plants, for this kind of traits. And of course, it could be also genetic manipulation with all the CRISPR-Cas things that are discussed nowadays in Europe, which is not really genetically modified, but
Could you maybe discuss some of the technical challenges associated with studying living plants?
yeah. So before going to the challenges, I just want to say that it's beautiful to work on living plants, because you work on multicellular organisms that you can follow in time with no very difficult rules to deal with, like not in the animal science. But the new challenges will be the time. So the resolution to look at it, because we are cell biologists, as I said before, and also the time. So we need to look at things in time, because everything changes according to the developmental phase you are looking at. Okay, so that's what is very nice, is that plants can root for a while, and at some point they cannot root so much, and so you have to follow everything in time, and that's in time and high resolution. This is really now the technical challenges we are facing.
So how does a work week look in hours? Do you need to work the 24/7? No.
Now, because we have automated microscope and things. So, but yeah, well, you need to work hard for sure.
Yeah, I see. Any more questions? Not right. Yes, we have questions on the third row, I think.
Yes, thanks for the nice presentation. I am wondering, because there is so much technology that is coming, there's more and more genomes of different plants that are sequenced, and we're working with more diversity. Do you see that? What is the future of Arabidopsis in plant science?
Well, I still believe this is still a good model, but I agree that we have to go to explore other ways. So like now in my group, we are also working on poplar, we work on lupin, we also work a bit on tomato. But the thing is that they work also differently. So if we want to really look at the basic things, Arabidopsis is still the king. I would say it's easy to transform, it's short in time for regeneration. If you compare to the work we do on poplar, for example, we face like a different time scale when you work on trees. So I think it's still bright, but we should consider always other species and understand how other species are also developing to learn even more. It's more knowledge accessible.
So you say Arabidopsis backtrav is the king, and you said something about why, but could you elaborate a little for those of us that are not into plant physiology and experimental things?
So
why?
It's a model. It's a genetic model nowadays, because from seed to seed you have 2 months and a half or 3 months. It's quite a short plant. I think full grown up it might be like this, so it doesn't take a lot of space in the greenhouse. It's fully sequenced. You can transform it with different constructs quite easily. So, yeah, it's like a better green yeast. It's a better green yeast. Yeast is also a genetic model.
Yeah.
But, yeah. So that's all the reason why they all, like in 2000 or even before, started with Arabidopsis.
I see. Yeah, and you mentioned in the video, and you talked a little about it in your lecture, the future applications that your research maybe could contribute to. I know you're into basic science, but anyway, could you tell us anything more about the more concrete ideas about concrete applications?
So like the fact that the cell shape is so regulated to maintain or create this tissue integrity is very important, because you need this tissue to be completely and take like good, so nothing can break it, right? So if there is wind, as I said, or if it's a bit cold or whatever, that the tissue stay good, so the plant is not damaged. And this is very important for the cell shape. The cell shape acquisition is very important for that, especially in the epidermis. But it's also the external tissue layer. But it's also very important for the function, of course. So this we could develop with this knowledge or select plants that are more resilient.
Yeah.
To understand better the way they could be more resilient.
Yeah, thank you. And, well, any more questions here? No, then I'll ask you a final question. Where do you see your research field is heading in the future, or will be heading in the future?
Ideally, or at least our group, what I would like to do in my group is really going to this diversity also on species. So like now we look at this cell, exactly same cell shape in poplar, and we see that it's a bit the function, the regulation of the cell shape is different. And how the evolution of this has been controlled, and how among the different species that are inside the water or outside the water, or how this is regulated to have at the end exactly the same goal, to maintain this tissue integrity. So that's where I would like us to go. For the field in general, I don't have so much imagination, but I think it will go well and fast.
Curiosity will tell.
Yeah, curiosity will
So
tell.
thank you very much, Stéphanie Robert.
Thank you.
Yeah. And now we will have a short break until 14:10, 2, and then we will meet Paul Cardol. So welcome back.